I'm Ian Braun. I'm a grad student at Iowa State University in Dr. Carolyn Lawrence Still's lab. And so I'm going to start by telling you a little bit about the tool that I'm building for my research, and then I'll move on to one potential use case of the tool, and then I'll briefly give an overview of the methods I'm using. So the function of the tool that I'm building is to take phenotypes that are encoded in text and convert them to an EQ statement format. And so what this looks like is we should be able to take a sentence like plants are shorter than normal, which could come from an online database or a research article, and convert it to a list of ontology terms where there's an entity, such as whole plant, in this case coming from the plant ontology, which is modified by a quality, in this case decreased height, which is coming from the phenotype and trait ontology. And once phenotypes are in this EQ statement format, they're easier to compute on, A, because they have this uniform E modified by a Q structure, and B, because they come from a uniform vocabulary which is limited by the ontologies that you're using to construct them. And then, of course, you also have all the other information that's explicit in the ontology from which you've drawn the terms. So one potential thing you can do with phenotypes once they're in EQ statement format is quickly calculate similarity between phenotypes even across species. And so I'll go through one example of how that might be interesting. Here we have two different mutant phenotypes. The top is a mutant phenotypes causing increased branching in neurons of mice. And on the bottom, there's another mutant phenotype causing increased branching in trichomes in Arabidopsis. And whether these phenotypes are represented in this image data or in the text from the papers where they're described, it's difficult to quantifiably say how comparable or similar the phenotypes are. But once the phenotypes are represented as EQ statements, calculating similarity is simply a matter of looking at the overlap between the terms that are present in the statements and all the terms that those terms inherit. So once you've declared, using this method, that two phenotypes are similar, you can hypothesize that they might share a similar or shared genetic basis. So if you know that gene A is causal to the phenotype in mice, for example, you can look for orthologous genes or genes in a common pathway in Arabidopsis. And a gene you find, like gene B, you could say is a candidate gene for the phenotype in Arabidopsis. So that's one potential example of how we can use this method to generate candidate genes. But how is the method actually working? So we start with a sentence like plants are shorter than normal. And the question we ask is, which ontology terms might represent the text? So what we're looking for is a subset of terms from all the ontologies that we want to use to describe the phenotype. And the method we use for identifying these is to use random forests which are specific to each ontology to classify whether the terms belong to that subset or not. Once we have this reduced set, we want to ask, how should those terms be combined to yield the best EQ statement? So we want to assign the roles of entity and quality to the terms that are present in that subset. And the method we use for this is to generate all the possible EQ statements you can form from that reduced set of ontology terms, and then probabilistically evaluate each of them with regards to a natural language processing analysis of the structure of the original text. So my official poster time was yesterday, but I'll still be around, and if you're interested in the project, I encourage you to come talk to me. I'd be happy to go into more depth about the methods and also share some preliminary results that I didn't have time to put in the presentation. I want to thank my other lab members, the conference committee, and my funding sources, the Predictive Plant Phenomics, NSF NRT, and the Plant Science Institute Scholars Program. Thanks.
Any questions? One question, please.
It's very interesting. Do you have the tool available?
Not yet, but we hope to soon.
Okay, thanks.